And so we have Dmitry Kolobkov. He did this work at GENXT, and it is examining federated learning within this application domain of genomics. And so I think this is a powerful, relatively untapped application area. I was super stoked to see someone working and diving into this area because I think Federated has a lot of potential for transforming the way that these models are built. And these models are right now being designed, and people are trying to figure out how they can um, integrate them into the kind of the existing network of data and companies and organizations out there. And so I think Federated is poised to really make an impact here. So with that, I hand over to Dmitry. If you, for those on the call, feel free next week, you're going to be able to meet Dmitry in person if you're at the Flower AI Summit. So over to Dmitry. Thank you for being here.
Yeah. Thank you. Thank you for inviting me here. And I would like to share the work of my team in federated learning on genomic data. So as Nick said, this is the area that would benefit immensely from federated learning, and yet the applicability of FL has been essentially not investigated. And we just recently published a study that that essentially explores the efficacy, the applicability of federated learning on genomic data. So I will provide the link at the end of the talk. So yeah, and of course it goes without saying that all federated models were trained with flower. So let's start. So every phenotype we develop, every disease, we it has some genetic causes and some environmental causes. Of course, there is a lot of interaction, and for most common phenotypes, like common diseases, the impact of environment is often more significant than our genetic predisposition. However, our genome has some important qualities, some important properties that makes it very convenient for us to predict the inherited part of our of the phenotype from genomic data. So unlike environment, we do know what our genome consists of, and like of course there are some blind spots, but generally we do know. We can also measure it. We can like for each individual, each individual can do a genetic test and obtain their genetic sequence that can later be analyzed. And another very important property is that our genome is permanent through our lives, and once we get the results, they stay with us, and this is why this genetic predisposition is not going to change. So how can we use it in practice? If we calculate this genetic susceptibility to a disease, like this picture, it's in the form of a polygenic score. So if we look at its distribution across different individuals, we can identify individuals with a higher susceptibility to the disease, and then we can monitor them more closely. We can assign some additional tests for them, and this is how we do not waste limited healthcare system resources on everyone, but we prioritize those who have higher genetic risk to the disease. And another important property of genetic susceptibility is that generally the dependence of phenotype from genotype is different in different populations. And this happens because of how natural selection works, and due to the fact that for many thousands of years, human populations were often isolated from each other and were evolving differently to different conditions. And this means that in general, phenotype from genotype predictive models, they're poorly portable between populations, which means that if we train a model on one population and test it on another population, then the results may not be great. And this leads to disparity in healthcare because most data in vast majority of the data in healthcare comes and in genetics in particular comes from participants of European descent. And if we apply these models trained on mostly European participants to patients from other populations, the predictions may not be great, and this may lead to poor decisions. So this is another additional challenge. So ideally what we would want to do is to train our models that are trained on data sets from all parts of the world. And this is just a subset of genetic data sets available out there. As you can see, most of them for people, most of the data here are from participants of European descent, but not only. You can see here Qatar Biobank, Taiwan Biobank, and so on. So what benefits would we have by training a single large global model on all this data, or at least on a part of it? So we would, of course, we would get larger sample size, and of course, this would lead, larger training data set would lead to more accurate models. And as already mentioned, we'll have more population diversity, which means that our models will be less biased, and also because we have larger training data set, we can include additional features such as rare variants and can investigate some additional phenotypes such as rare diseases, and we can get a lot of new insights. Right. So why do we want to use federated learning? This is mostly because healthcare data in general and genetic data in particular is extremely sensitive. And data custodians usually do not allow moving and merging the data. So we cannot just get access to all of the biobanks out there and download all of the data and train a single centralized model. Which creates an ideal scenario for using federated models. And so here we consider horizontal federated learning, and this is a cross silo setting. Right. So now we want to show the efficacy of federated models, and this is how we're going to do it. So we I need to mention that getting access to genetic data is usually not easy. You need to apply to fill the application form, then you need to wait, and so on. So it's really not easy to acquire a lot of it. So we managed to have access to the UK Biobank dataset. And what we do here is we take a single genetic data set, then we split it into nodes or clients. Then we train centralized, federated, and local models, where centralized model is trained on all of the data, all of the nodes, federated model also all of the nodes, but nodes are isolated, and local models are trained on a single node. And then we compare the performance. Of course, we expect to see that a centralized model is performing better than a federated model, but hopefully not much better. And we also, of course, expect to see that federated model is performing significantly better than a local model trained only on a part of the data. And because here we're especially interested in cross-population collaboration, we ideally would like to make our data split heterogeneous, so that we have high client dissimilarity, and this mimics the setting where we collaborate between biobanks all over the world. The problem is that with the UK biobank, there is not much population heterogeneity, and as you can see here, the data is mostly of participants of European descent, which is why we split this UK biobank data across data collection centers and not across ancestries. And of course, there is still some client dissimilarity, because these data collection centers, they are located in different parts of the country, and of course there is still some population diversity, but not much of it. And in order to test the models in a more heterogeneous setting, we considered another data set called 1,000 genomes, which is much smaller in terms of data size, but and it doesn't contain phenotypes, but it has much higher population diversity. So it is the study was conducted to represent different populations on the planet. So this is the overview of our two experiments. And so we predict phenotype from genotype on UK biobank, and we have 19 clients of very different size, and we predict quantitative phenotypes using lasso regression model, which is basically state of the art for this kind of task, and we implement Lasso as a neural network so that we can fully benefit from federated learning. And from prediction on 1,000 genomes, we predict one of the 26 populations. Also, this data is divided into 5 superpopulations, namely Europeans, East Asians, Southern Asians, Africans, and Native Americans. And again, there is a different sample size for the nodes. So, what are our results on UK biobank? So here on y-axis you see we plot test set R square for observed phenotype versus predicted phenotype. Like you see, like the correlation, the R square values are relatively low. This is because these complex phenotypes, they are dependent on genetics, but also there's a large impact of environmental factors. The x-axis represents different local nodes, and also there is one value for federated model and one value for centralized model. So we have 19 local nodes, and here we depict only 6 of them, not to overcrowd the graphs. And a single panel corresponds to a single phenotype. So here it's blood biochemistry phenotypes. And as you can see, blue dots, which correspond to a centralized model with centralized data preprocessing, performs best, but it does not much outperform green federated model with also federated data preprocessing. And these models much outperform local models with different ways of preprocessing the data. So you see purple and orange. And just as a matter of validation, you can see here that obviously, for local models, the more data we have on the node, the better the model performs, which is how we should expect it. And there's red is baseline, which is covariates only, and this is the model that does not include genetics at all. Right. So yes, here we basically see the relative performance of federated models compared to centralized and local. So now we want to investigate the more heterogeneous setting, and we take the 1,000 genomes data and split it by superpopulation. And as you can see here, this is a very heterogeneous split. So these 5 different colors correspond to 5 different superpopulations, and you see that they are very well distinguished by the first 2 principal components, which means that like this is the majority of the variance. So what we need to do here is to tackle the phenomenon that is called client drift. I'm sure many of you are familiar with it. So if we have heterogeneous clients, because of how federated model works, or because of how federated average strategy aggregates local updates, we may not converge to where we want to. This can slow down convergence and really lead to really bad consequences. So let's see how we can tackle that. Yeah. So before that, let's see that federated model does work here. This is a train loss on one of the 5 clients, and this is a federated average with 32 epochs in a communication round. So as you can see here, you see that every 30-second epoch we have massive increase in train loss. This is after we get an update from the server. Then the loss decreases as we train the model to local data, and so on. And the important thing is that the process does converge, though slowly, and you can see that because these peaks, they decrease. So yeah, so what you see here is like the upper border of this graph is basically these peaks. Right. So now if we change the number of epochs in a communication round, we can see that things are getting better. And this is the way of how client drift works. So as we saw, federated average with 32 epochs in a communication round converges slowly, but if we communicate with the server more frequently, leaving the total number of epochs the same, we converge faster. And this is essentially how it should like, and how it should look like. And this shows that in this setting, client drift can be successfully tackled by more frequent communication. Right. So let's see what we found and what are the limitations and the future directions of the future research. So first of all, we showed that federated learning indeed works, not only on a relatively simple task of predicting ancestry from 1,000 genomes, but also on much more clinically relevant UK biobank data. And we also noticed on 1,000 genomes that we can successfully tackle client drift by more frequent communication between clients and the central server. So one of the limitations is that this is still a proof of concept, because we took a single data set, like UK biobank or 1,000 genomes, and then we split it into multiple data sets. And in a real scenario where we take multiple biobanks, there are some additional challenges. For example, the feature space can be slightly different because different biobanks may have different genetic variants that are features in our model. And then some additional solutions have to be, additional steps of data preprocessing have to be made. Yeah, so talking about preprocessing of genomic data, this is a very a very important step, and it is not very straightforward for genomic data. You need not to you need to remove the noise, but not the signal. And this pre-processing, of course, has to be federated, because obviously there is no point in using federated learning uh, if you expose genetic data during pre-processing. So what else can we do? We all know that federated learning enhances privacy, but it doesn't guarantee it. So we can use some additional mechanisms. That really depends on the application. If we want to enhance privacy of single participants, then we can try using differential privacy, for example. And of course, we might want not a single global model for everyone, but we might also want to personalize this model to make it more suitable for a specific data set, while keeping all of the benefiting from the whole training data set. So these are potential future directions of the research. And finally, I want to thank people from GENXT, without whom this work could not have been possible. And there are two links. The first one is the link to our study, our paper that has just been published. And the second one is my LinkedIn. Please do add me. I'll be very happy to answer some questions now, but if you have more of them, please contact me. Thanks a lot.
Thank you so much. That was really interesting. Just as we did with the other talk, if people would like to post their questions in Slack or in the, let's see, the Zoom Q&A, or they can just raise their hand and we can take some of their questions. But while we're waiting for folks to ask something, I'll ask something. I was curious. I'm not so familiar with this type of task, and so I saw that you were predicting these superpopulations. Is that kind of the a conventional type of task that people who are exploring how you do modeling on this data often do? Is that like the MNIST or CIFAR 10 of genomics data building a model sort of thing, or maybe it's motivated
Yes,
by a
so
particular need?
right. So while it may not be as clinically important as predicting a phenotype, predicting participants, predicting clients' ancestry from their genomic data is an important product that genetic testing companies sell. So, their participants give their genetic data, and the companies return some insights, such as susceptibility to a disease, but also their ancestry that is inferred from genetics. And this can have it might be just only interesting for participants to observe, but also this data can to split in participants into cohorts and then doing some analysis on that.
I see. Yeah, your audio was just breaking up slightly there, but I think I could understand what you're saying there. That's cool. Interesting. I see. Again, if anyone wants to ask, happy to take them. Otherwise, I'll ask another one. I just want to double check one thing, and that was when you petitioned the data, that was based on there was some rationale for how the data was petitioned based on the location or the institute that gave the data. So it was it was a natural
Yes.
petition.
So basically, with the UK Biobank data set, participants there were given their data at different locations in the country, and there were 19 locations, and this is how we split the data.
Based on those 19. And was it a fairly even spread of data? Normally when that sort of when that's done, there's like heavy skews, one location or
Well,
few.
of course, like in more populated areas, there were more like higher density of these centers. For example, like I don't remember. I think there were only a couple of them in Scotland, and there were like more in southern England. But yeah, I think I would say that it's relatively fairly reflects the distribution of population in the UK.
I see. And did you need to handle that issue in any way, or do you just train just over it, irrespective of that skew?
Uh, you mean the fact that there are different number of samples in each node?
Yeah, there's imbalanced data in
Oh yeah,
the.
yeah. I mean, this is a very we wanted to see different sample sizes because this is a very realistic this is a realistic scenario. So like because of how federated average weighs the data, it's like this is just the strategy that we used, and it accounts for different node sizes.
I see. Cool, cool. I guess yeah. Actually, one question came in here. So they're asking, how much data would you need before you could train some of these more familiar architectures? So I guess by this question, they mean, I guess transformers and, all these other sexy neural architectures that people hear about. Do you need a lot more data, or is it, or perhaps it's not even to do with the data that they asked about if you need more data, but perhaps there are kind of other things that need to be understood before you can adopt the other models, or perhaps they don't make sense for the start.
Right. So there are different models have been tried on genetic data, and of course, with like now transformers are on the rise, and I really hope to see more transformer models for this kind of data. So we were using individual genetic variants. So essentially, the data was tabular, and I entirely admit that we lose a lot of information when doing this. But like this is how things are usually done in this field, and like there have been trained um, some CNNs and other more complicated architectures, and still as far as I know, the state of the art is a simple lasso regression. So there is a lot of a lot of features. So you do need some form of regularization for this. So we because we wanted to use federated model, we implemented lasso regression as a neural network.
See, makes sense.
I can see another question coming in via Q&A, and it says, are endpoints analogous to clients in FL?
No, I think that was about the last talk.
Oh,
Was
sorry. I just saw it now.
no problem. Yeah, no problem. But actually, there's one other one. I think maybe we're at the end of the time, but it's last question. It's a very natural question, from our community. They said this seems very cool, but how would they follow along? Like, can the data sets you use? You mentioned the UK Biobank. Are there other data sets you could point them to so that they could start to build similar experiments, or perhaps this is
Right.
completely regulated. They're kind of curious about doing work of this kind. It seems very cool, I guess, is what the main thing
Right.
about it.
So yeah, the problem with genetic data is that it's generally heavily regulated, and so this 1,000 genomes data set is publicly available, so you can just download the data and play with it. It does not contain phenotypic information, so it's basically just ancestry. Or like just these classes that you can predict, but it's still a very good starting point to get get familiar with the data. And with other data sets such as UK Biobank and like other biobanks, for example, you need to apply for access, and it's often given to research groups and companies. So
I
you
see.
need
So you
you
could,
need to fill in the application. It's going to be reviewed, and like you need to clearly state what you're going to do with this data, how you're going to use it. Then you need to pay a fee, and then if it's approved, you get access to the data.
oh, I see. I see. So there is some hope to do for folks out there to at least follow a little bit in your footsteps.
Yeah,
Interesting. Oh, great. Cool. Well, we've gone past our allotted time for Q&A. So yeah, thank you very much for the talk.